story. So what I'd like you to do is start off, tell us the hypothesis of your study, a little bit of how you tested that hypothesis and your results, and what you think the implications are. And then I'll ask you a couple questions.
Sure. So we were really interested in the role of alpha-synuclein in sporadic Parkinson's disease. So the hypothesis was that alpha-synuclein is an important pathogenic intermediary in the pathophysiology of the disease, and it's kind of difficult to test that because we don't really have great models of sporadic Parkinson's disease. And so the one thing that we've done that was unique is using this rat model. Rats which are intoxicated with Rotenone chronically develop features that look just like Parkinson's, including Lewy body formation in the nigra. And the hypothesis was that if we knocked out synuclein in the substantia nigra of these rats, that we could ameliorate the pathogenesis. So the approach we took was to design a gene therapy vector based on an AAV2 vector. It expresses a short hairpin RNA that targets the synuclein transcript. We showed that it very specifically and very effectively knocks out synuclein in the nigra when we do a single intranigral injection, and that in itself doesn't actually seem to cause any overt toxicity, either at the behavioural or the histopathological level in the rats. And then we took these rats where one half of the nigra was lacking synuclein and the other half of the nigra had normal synuclein levels but had been transduced with a control vector, and we treated them with Rotenone. And whereas Rotenone normally causes a symmetric bilateral nigral lesion with Lewy body formation and loss of dopaminergic neurons, in the rats that were lacking alpha-synuclein unilaterally, the side lacking alpha-synuclein was robustly protected, that they didn't develop loss of nigral terminals in the striatum, and they seem to have very significant, about a 30% more cells left in the nigra at the end of the experiment. And so we're kind of excited about those results because certainly it looks as though in that model, as though synuclein indeed is an important pathogenic intermediary. And that kind of tallies with a few things that are coming out of the sort of clinical, genetic and epidemiological literature. Firstly, it looks as though the genes in genome-wide association studies that are most closely linked to Parkinson's are synuclein and tau, and certainly some of the synuclein variants may actually affect the expression levels of alpha-synuclein. Secondly, that there's fairly robust epidemiologic evidence that links occupational exposure to Rotenone with Parkinson's disease in pesticide applicators. And so we think that this study is giving us some insight into the way in which the genetic predisposition and the environmental exposures might actually be interacting in vivo. The other reason we're excited, of course, is the potential for this to be therapeutically useful. Obviously there's a long way to go before one could test this in patients, but this is sort of proof of concept, at least, that you can knock this gene out in an adult animal, and it doesn't seem to do any major harm. But the animal then becomes protected against environmental triggers that we know cause Parkinsonism.
So when you give this viral vector, how much of the protein expression or the protein translation is knocked down?
Yeah, so that's another interesting question, and it's quite difficult to answer because the majority of alpha-synuclein in the nerve cell is at its nerve terminals. So right. you can't. There isn't a good way of quantifying it at the nerve terminals in the striatum because there are so many other types of nerve terminal there. Actually, if we quantify it within the cell body, we think there's about a 40% loss of protein, although there's very little synuclein in the cell body, so you're trying to quantify the difference between not very much and even less than that. But when we look at the transcript, it's much more robust than that. It's 80–90%, we think.
I see. So your short hairpin RNA knocks out the transcript, or does it prevent the transcript from being translated?
We think what it does is that it makes the transcripts get cleaved by the RNA interference silencing complex, and so it destabilizes the transcripts, and we can no longer detect the mRNA transcript.
I see. So, and is there a species difference, do you think, on our dependence or the necessity for having normal alpha-synuclein? Because I just happened to look up your paper in JBC on zebrafish, and it seemed to have kind of a different take on the necessity for alpha-synuclein, or was it a different kind?
Yeah, I mean, I think that's another really interesting question. Why do we have 3 different synuclein genes? And it seems certainly in mice, another mammalian species, that you can lose alpha-synuclein without any major abnormalities. In fact, you have to knock out all 3 genes in a mouse in order to get a very severe phenotype from lacking the gene. Some strains of mice have actually spontaneously lost their alpha-synuclein. As you pointed out, we showed that the same is true in zebrafish. Zebrafish have 2 copies of gamma-synuclein, and they've lost their alpha-synuclein. So it's not entirely clear, what we actually need alpha-synuclein for, if anything. But clearly that's an important question. If you were thinking about trying to knock this out therapeutically, I think we'd need to know a lot more about the potential toxicity of doing that, and a lot more about the normal function of alpha-synuclein.
And so with your viral vector, were you targeting one of the specific subtypes of alpha-synuclein then?
The viral vector just knocked out all of the alpha-synuclein, but it did that very specifically and didn't target beta or gamma-synuclein.
I see. It didn't hit the other synucleins.
That was the other synucleins, that's right.
Well, it's very interesting. I think this is very exciting. In the Rotenone model, when you use that model, does every animal have the same response, or how much variability is there in these kinds of studies?
That's another great question. So actually, when the Rotenone model was first introduced, it was extremely variable, and that made it very difficult to do neuroprotective studies. It turned out actually that the variability was because of difficulties with the delivery method for Rotenone. Rotenone is very highly lipophilic, and it's very difficult to keep in solution. The initial studies were using mini pumps to infuse the Rotenone chronically, and there would be a great deal of variability in how much Rotenone actually entered the animal. We've actually, Tim Greenemeyer's group, Tim Greenemeyer's the collaborator on this project who developed the Rotenone model. His group have now developed an intraperitoneal delivery paradigm where the rats get daily injections, and 100% of the rats develop a nibrostriatal lesion, and they develop it very, very predictably at around 12 days to 2 weeks of treatment with Rotenone. And so it means that we can use much smaller sample sizes, and it's much easier to do neuroprotective studies with this model now.
Oh, that's a big advance in that model system then. That's a big help.
That's exactly right.
Okay. Well, I think, is there anything else you'd like to tell us about your study? This sounds very interesting and exciting.
Yeah, I mean, I think the next steps, I think, are fairly clear. One is we need more information about the potential toxicity of knocking out synuclein. And we probably need to extend the studies into primates. I think we need to replicate the entire study and make absolutely certain that the effect is robust and reproducible. And then I think that looking at the mechanism whereby Rotenone and synuclein is interacting is potentially something that could yield novel targets for therapeutics, not necessarily gene therapy. And so that's one of the reasons we've been developing the zebrafish model, where we have transgenic zebrafish expressing human alpha-synuclein that becomes sensitive to Rotenone. And by using that model, and the tools that we can use in zebrafish for in vivo imaging, we're pretty hopeful of being able to get to the bottom of the mechanism by which the 2 factors are interacting.
One more question. So if you are looking at a larger brain, would you have to target the cell body or the terminal field with your viral vector?
Yeah, that's another great question. Actually, for this study, we did a single infusion of virus, actually just dorsal to the nigra, and it transduced about 90% of the nigral neurons. AAV vectors can actually flow by convection within brain tissue. They're so small. And so I think if we were thinking of this as a gene therapy, it would be an intranigral injection.
Right. Well, now with our experience in hitting near this area all the time with deep brain stimulation and putting electrodes there, it's not as a daunting a thought as it would have been 15 years ago.
I think that's exactly right. And actually, we're also with the experience of using AAV vectors in Parkinson's clinical trials, actually transducing the brain with this vector system isn't such a daunting prospect either.
Right. Well, that's been Ed Burton from the University of Pittsburgh telling us about his new and exciting study that he's presenting at the American Neurological Association annual meeting in Boston. Ed, thank you very much.